This short webinar is the fourth in this series, dedicated to explaining various aspects of the MMRRC strain GQC report. It focuses on a special collection of 63 multi-parent recombinant inbred strains known as the collaborative cross, or CC, population. The GQC reports for CC strains differ from the standard MMRRC reports in several key aspects, which we will review here. The first reason for these differences is the fact that the genomes of CC strains are derived from 8 inbred strains, and thus representation of the genome in terms of primary and secondary backgrounds is meaningless. The strain GQC report is not intended to be a substitute for the 8-way mosaic that defines each CC strain. The second reason for report differences is that most of the CC strains are not yet fully inbred, and importantly maintained as live colonies. Therefore, residual heterozygosity will decrease over time. In addition, the main purpose for continuous GQC in these strains is detecting and avoiding propagation of rare breeding mistakes. These factors underlie the decision to represent the genome of CC strains in terms of regions that are fixed for a given CC line, shown in black in the ideogram, and regions that are still segregating, shown as orange in the ideogram. The genome analysis section describes the genomes of CC strains in the same terms. In contrast to other MMRRC GQC reports, the report for CC strains makes no attempt to estimate recipient, donor, or tertiary backgrounds. In addition, no locus and allele of interest are present in the name, summary, or ideogram, as these features are irrelevant for the collaborative cross. CC strains are maintained as live colonies at UNC and are subject to regular mini-muga genotyping. The MMRRC at UNC uses these genotypes to update strain GQC reports approximately every 18 months. This is reflected in 3 ways in the strain GQC report. First, the date of the report will change over time. Second, the number of samples genotyped is typically much higher, as they represent the entire complement of breeders at that time. Third, the fraction of the genome that is segregating will decrease over time. For example, compare these 2 GQC reports for CC36. The one on the left involves 15 samples from 2020 and has 4.3% residual heterozygosity. The report on the right was based on 24 samples genotyped in 2024, and residual heterozygosity has reduced by more than half. MMRRC customers benefit directly from up-to-date and accurate reporting of the levels of residual heterozygosity present in strains that they plan to use. It is important to note that small regions of residual heterozygosity may not be detectable by mini-muga, and thus are missed in this analysis. The report summary notes this point. The CC GQC report has additional unique aspects, including the identity of the founder strains present in a given CC strain. Finally, unlike other MMRRC strain GQC reports, no mention of diagnostic SNPs is included in either summary, nor in the dedicated diagnostics section at the bottom of the report. This is because 3 of the CC founder strains are wild-derived in origin, and diagnostic SNPs can only be reliably scored in the context of classical inbred backgrounds. Additional webinars will be created and posted on the MMRRC website. We would like to thank our funding sources and everyone involved in the MMRRC strain GQC project.